Hello, my name is Isiksacan. I'm a postdoctoral fellow at Harvard Medical School and Massachusetts General Hospital. Together with my colleague Eda Aşık, we have prepared this video describing the red blood cell osmotic fragility test. We can categorize the solutions as hypertonic, isotonic, and hypotonic. If a cell is placed in a hypertonic solution, there will be a net flow of water out of the cell, and the cell will lose volume. If a cell is placed in an isotonic solution, there will be no net flow of water into or out of the cell, and the cell's volume will remain the same. If a cell is placed in a hypotonic solution, there will be a net flow of water into the cell, and the cell will gain volume. Red blood cells are happy in isotonic solutions. The osmotic fragility test is used to measure red blood cells' resistance to hemolysis when we expose them to solutions of varying salt concentrations. We prepare 12 different saline solutions with decreasing salt concentrations. At the highest salt concentration, we have 0% hemolysis because the solution is isotonic. As we decrease the salt concentration, we expect that cells swell and hemolysis increases and eventually becomes 100% at zero salt concentration, which is actually the eye water. When hemolysis takes place, cells release their internal content, namely hemoglobin, into the supernatant. Absorbance measurement of the supernatant quantifies the hemolysis level and hence osmotic fragility of red blood cells. Mean osmotic fragility is expressed as a salt concentration where we have 50% hemolysis. We first prepare the stock saline solution. We dilute the stock solution to prepare a working saline solution. Then we prepare 12 buffered saline solutions of decreasing salt concentrations. The lowest concentration corresponds to DI water. After the buffered solutions are prepared, we mix these solutions with packed red blood cells and incubate them for 30 minutes at room temperature. Following 30 minutes of exposure to osmotic stress, we centrifuge red blood cell samples to collect supernatants and pipette them in a 96 well plate. Finally, the absorbance of supernatants at 540 nanometer is measured to calculate the hemolysis levels and therefore the mean osmotic fragility of the sample with respect to solution concentration. Now, you will see the assay in action.